So I'm going to speak about a fungal pathogen, Aspergillus fumigatus, and look at the development of potential therapeutics for treating this pathogen. Many of you will have seen the series, the beginning of the year, The Last of Us, which looked at fungal pathogens that took over people's brains and destroyed civilization. So that's science fiction. This is science, but the fungus is, as you'll see, quite a serious one to be dealt with. So Aspergillus fumigatus, as I've said, is a fungus. It's widespread in the environment, and we come into contact with it on a daily basis. We inhale conidia or spores into our lungs. For the vast majority of us, this isn't a problem because our immune systems are set up to deal with it. But where our immune systems are defective or we have underlying disease, then it becomes a serious pathogen. And it causes a wide range of diseases, from colonisation to invasive diseases. And the most serious form, which is invasive aspergillosis, kills around 600,000 people a year, which is actually more than the number of people who die from malaria. So it gives you an idea of how serious it is. The World Health Organization at the end of last year did an analysis of all the fungal pathogens that affect humans, and they broke them down to different categories. And Aspergillus fumigatus is one of the 4 most critically important pathogens. So it's a serious threat. So as I said, our immune system is set up to identify these conidia when we breathe them in. So they're identified as they germinate, and then they're eliminated quite rapidly. So in the space of about 2 to 4 hours after inhalation. However, where there's a disease such as cystic fibrosis or asthma or COVID or another disease such as leukemia or transplantation, the immune system is impaired. So the conidia germinate and begin to spread through the body, and then in the worst cases, they develop into systemic aspergillosis, which has a mortality rate of around about 80 to 90%. So just to give you an idea, this is the fungus that we breathe in. We breathe these in. The immune system doesn't really see these before they germinate, but as soon as they germinate, which takes about 2 to 4 hours, once they be into this form, the immune system sees them, identifies them, and kills them. Then, if the immune system doesn't work, they continue to germinate and grow, and these black threads, that's the fungus growing through pulmonary tissue. And depending on the level of immune suppression or immune dysfunction, the fungus continues to germinate and grow, disseminates through the body, and at post-mortem, you'll see fungal colonies lining the inside of the lung. So it has become that systemic. The transition from inhalation to death in susceptible patients is typically about 5 days. So antifungal therapy has to go in very rapidly to arrest this development. There's only a 5-day window. So what therapies do we have? We have a range of therapies that are used. There's the azoles, which interfere with the synthesis of this sterol in the fungal cell membrane, called ergosterol. They're widely used, but in the last 10 years, Aspergillus is developing resistance to these. There's another group of drugs called the polyenes, and one of these is called amphotericin. This binds to ergosterol and punches holes in the membrane. Polyenes work quite well, however, they're extremely toxic. And patients who are on prolonged polyene therapy are often cured, but will then need dialysis or kidney transplants afterwards. So toxic is it? So they're not an ideal solution. So the project that I'm going to describe to you has 3 strands, which cover 3 themes within SSPC. The first was to develop novel gallium complexes, and this was led by Darren Griffith. And then we want to evaluate these as novel antifungals. Would they work against drug-resistant Aspergillus fumigatus? Would they provide a different mode of action to existing antifungal drugs? And this work was carried out by myself and Anne-Marie Healy with collaborators in the University of Pisa. And then the third aspect of the work, which Anne-Marie Healy has led, has looked at formulating these novel complexes with a view to potential therapy. So we're using gallium as our metal. And why use gallium? Well, gallium has been used in human therapy for many years. It can be well tolerated. And interestingly, it is an iron mimetic. Microbes see it the same way they see iron. And microbes have an absolute requirement for iron. When they get into the body, in order to grow, they need to get iron. And where would you get iron in the body? Well, it's in hemoglobin. So they steal it from hemoglobin, incorporate it, and then they can grow. So they have an absolute requirement for this. But because gallium is similar to iron, if we put in gallium, they take that up, incorporate it into proteins, and then they don't work. So in this way, we might be able to hijack their own biological systems. So we looked at the ability of gallium. We looked at a number of gallium complexes. One in particular was gallium maltolate. And we evaluated the ability to inhibit the growth of the bacterium Pseudomonas aeruginosa. And we found gallium maltolate is very effective, and it interferes with it induces an iron-limited stress response in Pseudomonas. So this metal, gallium, works against a drug-resistant pathogen. The next part of the work then, Darren suggested, was that we should weaponize gallium. So we know that gallium has antifungal, antimicrobial properties, but could we make it better? Could we attach to it this siderophore moiety? And siderophores are produced by microbes, by bacteria and fungi when they infect the body. And siderophores are used for catching iron. So they catch the iron, take in the siderophore and release the iron internally. So it's an iron capture system. By sticking this onto the gallium, we could be able to fool the pathogen, like a Trojan horse. It wants the siderophore because it thinks there's iron attached to this. I need the iron, so it'll take in the siderophore, and then it releases the gallium, which it still thinks is iron, and then incorporates it, and this results in death of the cell. So these were the complexes that we were interested in evaluating. Would they work? The synthesis of these complexes is described in this publication. And as part of this publication, we tried out and examined whether these complexes worked in vivo. So to do this work, we used insect larvae or caterpillars, which have an immune system very similar to our innate immune system. So we can infect them with a pathogen, and we can follow up with a treatment. And they respond the same way as a mouse would or a guinea pig. So in this case, we infected them with a pathogen, Pseudomonas aeruginosa, and then we dosed them with complex 1. And we show in vivo activity because we could get increased survival of the larvae compared to the controls. So the complex works in vivo, and we get survival of the host. So encouraging in vivo data. So to do our work, we wanted to evaluate whether this complex would work against Aspergillus fumigatus. Was it more effective than the existing complexes, the pre-existing complexes, and how did it work? Does it work the same way as azoles, polyenes, or does it work in a different way that might have application for dealing with drug-resistant Aspergillus? So the first thing that was done was to carry out a toxicity assay. So this is where we expose Aspergillus fumigatus to increasing concentrations of our different complexes. These are the complexes, the siderophore complexes we're interested in. These are the salts, the pre-existing salts. And what we can find is where we use the siderophore complexes, the ones that Darren made, these are highly effective and they inhibit growth quite rapidly. So they stop the growth of the fungus. They also have the ability to kill the fungus. So that's good. So now we have something that we've demonstrated previously has some in vivo activity. And now we have in vitro data that it inhibits Aspergillus fumigatus from growing. The next part of the equation then was to figure out how it works. And to do this, we use a technique called quantitative proteomics. And this looks at the proteins in the cell. So every cell contains DNA, RNA, carbohydrates, sugars, lipids, and proteins. And proteins are the molecules that do the work in the cell. They're the functional things in a cell. So if we can identify the proteins in the cell, we can see if their abundance increases or decreases. How do they change? And quantitative proteomics allows us to do this. So in this case, we took our Aspergillus, we exposed it to our complex, we extracted the proteins, and then we chopped these up with trypsin, cleaned them up, purified them, and put it into our mass spectrometer, which identifies the proteins, sequences them, and then calculates the relative abundance. Is there more of the protein or less? And this gives us an insight into how the cell is responding. And we can use a variety of data analysis packages to interpret the data that we get. But the whole program here is basically giving us an insight, how is the cell responding? And we get a lot of data, and just two things to show here. We identified 2,400 proteins, and of that number, 850 were changed in abundance. So 850 proteins changed. A lot of proteins weren't changed because they're not affected. They're just doing regular things, but some proteins are changed. One way to analyze this data is to do this principal component analysis, which gives a global overview. How are the cells responding? Well, we can see this is our siderophore complex treated cells. They're down here, quite distinct to the controls. So there's a big difference. There's something going on. It's sort of a big change. Here's our gallium nitrate treated cells. They're showing a bit of a change relative to the control, but the siderophore complex is giving us a big change. So there's something going on. Another way to analyze this is by a heat map. And this shows the proteins that we've seen, and the color tells us whether they've increased in abundance or decreased in abundance. So this is our siderophore complex treated cells, and we can see a load of proteins here are increased because they're red relative to what we see in the control. So a number of proteins at that level in the control are really ramped up in response to the treatment. And then down here, a number of proteins are decreased in abundance relative to the control. So again, it's telling us there's something going on here. We can present this data in a variety of ways, and this is a volcano plot, but it just shows us when we treat ourselves with the gallium siderophore complex, we get this distribution. Some proteins up here increase, some proteins decreased. But just to look at the scale, this is a log scale. It's a log 2 scale. So 2 to the 2 is 4. 2 to the 8 is 256. So there are some proteins in the cell, in response to the treatment, are increased 256 times plus. So there's a massive ramp up in those. And they're the ones that are responding. So they tell us something about the treatment. So what are these? Well, we can identify some of them. These are the relative fold changes. And we see, for example, increase in a protein called Kupin domain protein, which is involved in the stress response. So the cell is under stress. Well, that's good. We see an increase in ergosterol, the cell trying to protect its membrane. We see an increase in siderophore biosynthesis. So the cell is trying to make siderophores because the ones it's bringing in aren't working. It's not giving the iron it needs. And there's also a decrease in the abundance of certain proteins. And some of these are associated with the mitochondrion, some of which is the energy generating component of the cell. Some are involved in protein translation. So this begins to give us an insight into how the cell is responding. But we can look at it at another level, And the other level looks at pathways and processes. And this is called a string network analysis. So what this does is it looks at all the proteins that are changed in abundance, increased or decreased, and puts them together into groups. So when the cell is exposed to our gallium siderophore, we see an increase in carbohydrate metabolism, we see an increase in oxidative stress, we see an increase in DNA repair. The cell is being damaged. We see some fatty acid increased. So the cell is responding by increasing the abundance of certain proteins. On the other hand, it's also decreasing the abundance of certain cells or certain processes. So we see here proteins associated with the mitochondrion are decreased in abundance. The cell is shutting down or closing down the mitochondrion. Is this the target of our treatment? And other processes are also being reduced. So the mitochondrion is reduced. Is this effect an effect of gallium or our siderophore? And to do this, we carried out a similar type of analysis with gallium nitrate, which doesn't have the siderophore attached, and we get a distribution like this. And then we can look at the proteins, and we see similar proteins increased in abundance, similar proteins decreased in abundance that we saw with our complex. So it's not the siderophore that's causing this effect, it's the gallium. The siderophore is getting the gallium into the Aspergillus better. That's the trick. And as I said, the gallium-treated Aspergillus fumigatus shows reduced abundance of proteins associated with the mitochondrion. And the mitochondrion in a cell is responsible for respiration, for energy generation. And we can measure respiration in a cell. And this is measured using a compound called tetrazolium chloride. And we can see that following treatment with our siderophore complex, there is a reduced chemical reaction, which is proportional to a reduction in respiration. So the mitochondrion is the target of the gallium, we speculate. So what can we say about this complex? Well, we know that gallium has antimicrobial properties because cells take it in thinking that it's iron, incorporate it into proteins and other constituents within the cell where it doesn't work like iron, and then those functions don't occur. So the siderophore, complexing it with a siderophore, allows us to get it into the cell faster to have a bigger effect. So this then, we know the cell responds by increasing siderophore biosynthesis because it wants to make more of these. There's an increase in DNA repair. There's an increase in the stress response of the Aspergillus. But it disrupts respiration. Why? Because cytochromes, which are essential for respiration, have iron at their core. If the cell can't get iron to make cytochromes, it can't grow, can't develop. So what we've been able to show then is the gallium siderophore complexes are more potent than the existing formulations. They work against Pseudomonas aeruginosa. They work in vivo in Gallium larvae, in the caterpillars, and they work in a different way to the existing drugs, to the existing azoles and the existing polyenes. So when we're working with an Aspergillus that's resistant, how can we treat it? One group of drugs it doesn't respond to, the other group, they're too toxic. Now we've some potential by weaponizing gallium, that we can hijack the cell, get the complex into the cell where it can disrupt respiration and so reduce the growth of the pathogen. So before I finish, I would like to thank the people who've worked on this project. Magda and Brunella did all the proteomic work that I spoke about. Lewis did the synthesis, and also my colleagues Darren and Anne-Marie and Karsten, and also our colleagues in the University of Pisa who we've collaborated with. So thank you for your attention.